at night. I admit I feel the same, facing my faults every day. I'm draining blood just for a taste. I can't replace times like these you fall apart, and it's times like these that make you who you are. Breaking down, so can you tell me now, was it worth it? Do I deserve it? Our time is running out, and now I'm left to drown. I hope it's worth it.
Hello. I actually really prefer like hard rock or metal or like alternative rock or whatever, but I just feel like for stream starting with that is just not the energy I personally bring, so that's why I always choose like that sort of like lo-fi or whatever it's called. Yeah, lo-fi hip hop beats. Yeah, welcome everybody. Micro, yeah, I hope you're okay. Yeah, of course it happened when you had things planned. That's annoying. It's not COVID, is it? Let's see, who else is here? Brian's here, Alt Miss Frizzle, Managard. Welcome everybody. Okay. Let's see. Your hair may give it away these days, but you also like Zemetin. What kind in particular? I grew up on like Rammstein, and that has very much affected what I like to listen to. Although I like just about anything. I've recently been listening to Ghost. Hold on, Ghost? Yeah, these guys. They're good. I'm also very picky, not because I think I have better taste than anything else. Just, I don't know. I just listen to half the songs I hear that are popular, I don't like, and I don't know. There's not like a through line between any of them. Your rapid antigen tests say no. Well, that's good. Hello, Benjamin. Yes, new weird pond stuff. Did I say weird? Oh, I didn't. I actually meant to say that. Well, we're on the same wavelength then. Yeah, pond stuff. Speaking of which, I'm going to turn that on. I had found something right before stream that I was hoping not to scare away. So let's see. You're editorializing? Well, you're absolutely correct. Your dad would listen to Rob Zombie when you were a kid. Love Rob Zombie. Then a lot of other 80s bands. Your preference was like punk of the mid to late 2000s, listening to Attack from high school sometimes. I feel like we have overlap, not dissimilar music interests. Alright, where is the thing that I found? Which is a super, descriptive way of putting it. I think that's it. See, I think I found a ciliate, which has a fun little attachment bit to it. Yeah, look at you. Still there. I didn't want to scare him away. Oh! Look at that. Oh, look, you can even see... Yeah, you can see how the little propellers on the side of its, I'm going to say face, are moving things around. I'm going to take a screenshot. Yeah, exactly. It's attached to whatever that was. I was more afraid of like the heat of the microscope kind of making it unhappy. So I had the light off after I found this guy. Yeah, so I've seen Diatom's attack. He finds these things all the time. I have just not been lucky enough to find one. This came from a pot. No. So the bottom of like a flower pot that you take off, It's been sitting on our deck just without a pot on top of it, gathering all sorts of like dirt and detritus. It was turning strange colors, so I figured something must be growing in it. And yeah, so I have some little, sorry, that was from me moving my my stand. I'm actually gonna go turn the music back, only because I don't like the hearing words in the background, because it is a little louder for me than it is for everyone else. Oh, what was that? I clicked away and I just came back and something huge went by. Hold on. Where did that go? What was that? Yeah, a new friend appeared, exactly. What was that? Microscopic jump scare, exactly. Oh, 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 oh. Whole bunch of them. Oh, look at that. They have their own little ciliate mouths going on. Come on. I think this is what it was, because I was a little bit more zoomed in. Yeah. These, well, they're not going to behave, though. They're going to be zooming all over the place. Wow. Yeah, so this big boy crossing the street. Thank you. That also reminds me, if y'all ever clip something and you feel like it, I'd super appreciate if you linked to the clip in the clips channel in the Discord. No pressure, but it's there. And I'm attempting to work on a script to do that automatically, but it's been a little finicky. All right, so here's some cool stuff. What's going on? Oh, there they are again. Okay, I think we're just going to continually see those guys. I want to see what these are. Yeah, so this sample is more or less just a gross, dirty puddle sample. And then I also have a more or less fresh sample from a lake, a local lake. Not like the pond that I usually grab from. This lake, although it wasn't dangerous now, apparently about a month ago, a dog died because because of the cyanobacteria like infestation that occurred. And they closed off the lake for a good part of the month. Oh, are we back to our little ciliate friend? I think we are. Went all the way back. All right. So what I don't. Oh, that's good. I just gonna want to watch him. I think your speedy friend might be a Hypotrix? Let me look him up. Whoops. Based on the big cilia bundle. All right, I will trust you on that. And trust but verify. Hypotrix. Oh, yeah, that looks about right. I think you're right. Let me switch quick. Yeah. Hypotrix ciliate. I mean, yeah, I think you're right. Absolutely right. Cool. I wish they would slow down though, so we could get a nice view of them, but I don't think that's actually going to happen. Same thing with this guy. I'm willing to try, but I'm sort of afraid of it. Trying to just take this and putting it under a slide so I can use my 40. Yeah, my 40x objective and get a really good picture of it. I think that would also flatten it out a little bit more. Hello, David. Welcome. Just checking out some mud sample. Not mud. What did I call it before? Just dirty puddle sample. Yeah, I think I think I'll try it. It looks like it's attached. Excuse me. I think it looks like it's attached just to the bit of like detritus that's kind of at the bottom part of the screen. I think if I just very carefully pipette that, I can get it under a slide. The slide will also be better because then it's less susceptible to like rocking when I touch, literally any part of my desk. Just gotta find a slide now. Also, sad news for anyone following the saga, but the caterpillar has died. It did not survive its wasp nor viral infection, unfortunately. It was pretty much done for. Good news is I found another one. And all the wasps eclosed. So all the wasps came out of their pupa. I let most of them go. A good portion also didn't eclose, which is kind of sad. Yeah, pour one out for the homie. Yeah, he died. Yeah, a good portion of the wasps didn't eclose. It's kind of strange because the pupa turned... So when insects eclose, that's the word for leaving the pupal casing. When they do that, they often turn darker right before. So that kind of lets you know that they're about to come out. And a lot of them turned dark, like they were going to eclose, but then they just never did. And now it's been several days after the main batch kind of went off. So I'm not really sure what went wrong there. I'm just going to cut the tip. He died as he lived eating. That is actually true. It was very sad. He couldn't move anymore, so I kept putting new fresh leaves in front of its face and would kind of eat like a little circle around it because its like front parts could move. But then it finally died. And then, yeah, super big coincidence, but someone who works in my lab now used to work on that particular type of larva, that particular type of wasp, and that particular type of virus. And they actually tried to work towards figuring out how they could like save the larva. Whoa.
R.I.P.
Yeah. R.I.P. indeed. Thank you, Brian, for the resubscription. I appreciate it. Yeah, so they worked on this problem, and as it turns out, they couldn't figure it out. So I thought my clever idea before I talked to her was, what if we like inject it with ecdysone, which is the like hormone that insects use. They get a big spike of it when it's time to go into like a pupa state because they basically just get stuck. The virus paralyzes them and prevents them from pupating, after which they could survive. But it turns out they tried exactly that. It doesn't do a thing. I also learned a fun fact. These larvae... Sorry, I'm just watching what's on the screen now. These larvae are also... Oh, and sorry. When I say larva, I mean the caterpillar. They're a larval form of the moth, but the caterpillar, these caterpillars, they're screwed from the beginning because apparently some of the plants that they feed on, like tomatoes, they release air, like certain compounds that help attract the wasps. So the thing that they're just trying to eat, which to be fair, defense mechanism, releases these chemicals into the air, and then the wasps smell that, so they know where the caterpillar is. They go and lay their eggs in the poor caterpillar. Inside the ovaries of the wasp live this virus, which exist to get infected into the larva. Now, they don't actually replicate in the larva. The virus doesn't actually do anything like replication wise in the moth larva. Instead, what they do is they paralyze them and shut down their immune systems so that the wasp larvae growing up inside of them have an easier time. And then, of course, the virus, the thing they get out of it is that they infect those wasp larvae and, live on for another generation. Yeah, it's all connected. It's crazy. One of these crazy systems. All right, I got to zoom out a little bit if I'm going to try and grab this ciliate. Oh, I've made a poor choice. Oh no. Where did you go? Oh, Brian, I see you contributed to the community challenge. I was just trying it out. So feel free to contribute and I'll do it if it gets to it, but I also just was trying to figure out what it is. Yeah, I just have a bunch of random packs of cards that I can open on stream, but there's no particular reason to do so until I get a reason. It seems to you as if moths don't cling to life a lot anyways. Many moths that fly into your room at night end up dead either on a random shelf or in the glass of water. Ooh, you didn't finish beforehand. Yeah, a lot of moths, well, some moths also aren't born with the capacity to eat, or at least eat much. So their lifespan is basically as long as they continue to have energy. I mean, that's just how evolution works, right? The moths that do make it and to go off and reproduce, they'll make hundreds of babies. Some have to survive, right? At least that's their thinking. Okay, sorry about this. I need to hold down this thing and then go back. Come on. I am failing at this so bad. Moths around you usually end up as a neat little snack pack in the spider's webs. Yep. Where's my pipette? I might have gotten it. All right. That's so that's why they seem to come into your room to die. Thanks. I think it's actually because they navigate via the light from the moon and basically every other artificial light. They see and they're like, oh, moon. And so they attempt to travel towards lights or something like that. It's something about the moon and not knowing how to navigate. I mean, they're not smart creatures, right? They're just insects. I feel like we do kind of think of moths and butterflies as like weirdly smart, but of course we discriminate. So we think, butterflies are beautiful and smart, whereas moths are drab and dumb. But, they've probably got about the same size brains and all that. All right. Let's see if I actually managed to get something. All right. Actually, no. Yeah. Just insects. Well, I can say that I work on insects. Insects, they're super efficient. They're good at their jobs, but you know they're not going to win any. They're not going to win any awards in terms of like intelligence. They're really good at making more insects, though. See, I zoomed out. Now I need to zoom back in. All right, so it was attached to one of these. Is that it? No. I just have to find the little bit that it was attached to. I hope it didn't just fly to the edge like most things that I try to mount. I should really like take a class in how to actually do microscopy or something like that. You stood up to get your food delivery and almost passed out from standing up fast. Oh, and you're also nauseous. The only insects that matters are dragonflies. Dragonflies are pretty cool. I do love a dragonfly. They're a pretty old clade of insect too, aren't they? If I lost the ciliate, I won't be surprised, but I'm going to be sad. I really wanted to see it up close. Come on. Yeah, old and hyper efficient. Yeah, they have like a, I was about to say KD ratio, but I guess it's sort of like accurate. They kill like a huge proportion of the targets that they take. Like when they decide to like target an insect, it is very, very likely that they're about to kill that insect. You know, that's not necessarily true for everything. I think like mammalian predators are quite inefficient if you compare them to a dragonfly. It's 90+%. Wow. I think I lost the ciliate. Sad. Well, that's okay. We can, I'm just going to examine this just to make sure. This didn't look like the bit that it was stuck to, though. I didn't really see any of the stuff that it was on in the slide, so I wonder what happened. This slide also is dirty? Well, that's great. Super helpful. It can't be dirty. This is a new one. Oh well. I think what I'll do, well, I don't want to monopolize this. We can either go back and look at the same one or go to the pond one. Let's look at a little bit more of what we already have. I'll grab a little bit more from the jar sitting next to me. Maybe I'll start by putting it under a slide, actually. That might be a better use of time. The secret is that they are able to predict where prey will be. Really? Dragonflies are so cool. You mean like they can anticipate where their prey is going to go? Or like they're like, oh yeah, that tall shady grass over there, that's where my favorite meal is going to live. Because they're quite what's the word? Dexterous, but for flying, because they zip around a lot. Ah, the first. Okay, I see. Let's see. Which one of these was it? Ugh, gross. All right. This water is also disgusting. It's the color of like tea, except the tea is from all the rotting plant material. They talked about it on QI, really? But they talk about everything on QI. But QI recently? Like, would this be something I'd be able to find kind of easily? I love QI. There's some like thick things on here too. All right, let's see what we got. One good thing about being sick is that eating takeout all the time is perfectly valid. Absolutely. That's something we don't talk about enough. It is perfectly valid. You had a rose bush that was dying when you moved it into this house in July, and now it has 5 blooms on it. Oh, nice. Ooh, here's a bit of that rotting plant matter that I told you about. Maybe something's living on it. There's a surprising amount of depth. I think something in this is kind of thick. What's this? Oh, I found one. Look at that. Just right there. Perfect. What's it attached to? Oh, it's... oh no. Its attachment point is a little small for its propeller force. Did we talk yet about No Such Thing as a Fish podcast? It's the weekly podcast by the researchers of QI. 4 vaguely related interesting facts of each week. No, we haven't talked about it. I was just about to bring it up. No Such Thing as a Fish is probably top 3 for podcasts for me. I love it so much. It looks like a stumpy-legged head crab. It does. Oh, wait. Was that one that just flew by? Like, just a stalkless one? It was about the same shape. Okay, I'm going to zoom in and attempt to check this out. There's a little bit of depth, so unfortunately it might not work out. Please, 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 please. It disappeared. Where'd you go? I lost it. Or is... oh, no, I didn't lose it. It was right there. That was its attachment point. Yeah, please. Everyone should check out No Such Thing as a Fish. I mean, it's so good. If you like facts. Also, look at this little guy. Oh, he's moving around a lot. It's going to be hard to keep up with. Oh. Did he just catch something? He just... oh. Whoa. What's he doing? Is that it, like, eating? Very cool. I like that he has little... That's so fast. It almost like looks like frames are being skipped, and it's like eating animation. Ologies is also a very good podcast. I don't listen to it as much as I should, but I'm very much aware of it and have listened to a bunch of it. Did an episode on critical ecology. Okay, I'm just going to go ahead and do that. Now he is making it difficult by moving in and out of focus. Oh, that one was my fault. That's my bad. Stay in focus. Just trying a little bit of a filter. That doesn't help one bit. All right, this guy is kind of on the move. Very nice. But I think I'm going to, we'll look around a little bit more, see what else is on here. There's a bunch of stuff on the slide, and it'd be a shame to like waste it as it's drying out. I really don't think they're meant to do it like that. Because these guys, whatever they're called, I had the name up at some point, but I don't remember. Yeah, they stalk themselves and attach themselves down, but they are pretty mobile without their attachment. All right, let's go look around. Bunch of little things right there. What's going on in this neighborhood? Pretty timely because there was a study released about how climate change made Hurricane Harvey worse specifically for low-income neighborhoods. Interesting. That's, well, interesting, but also extremely distressing. And it kind of makes sense just because of how the economics of that and the pressures are going to work. Like, of course, in any city, the lowest value property is probably only going to be the stuff that is more susceptible to natural disasters. And there's not going to be any broad investment to fix that. Are we looking at like an amoeba or something that we can't see the edges of? What is going on here? It seems like there's a... It seems like this mass of moving things are kind of moving together. Maybe it's just something sticky? Weird. Maybe I'll go to the high magnification already. So that's something I haven't definitely seen. I have not definitely seen an amoeba floating around. I have seen them before, and I remember them having kind of distinct outer edges. It's just kind of weird. It just kind of looks like a bunch of things, little guys that are like stuck in something, but that something is moving. Huh. Here. So I will zoom in on that though, just to see. Zooming in. Ah, there we go. Doesn't help that my lenses are scratched and or just dirty. Yeah, that doesn't really make it any more clear, although we can see what those things are. Oh, there's a couple different types in there. Del Maximum had these weird jelly blobs that end up being Ophiuridum. Let me look that up. Let's go to main. If I can find where my mouse is, let's turn that on. Ooh. Well, I don't want to move things. Ophiridum. I think I spelled that wrong. Oh, interesting. Little algae? Let's go to all. Colonial animal that is a green algae. Huh. Are they freshwater? Just because this would be freshwater. This jelly with all kinds of ciliates, rotifers, etc. living inside. Interesting. We'll keep an eye out for that. His was found in a lake. Yeah, this isn't even my lake sample yet, but who knows? So now we have a little green guy, but it kind of looks like he just got stuck. Is it still symbiotic if they just get stuck? Probably not. You can see those little like needly looking things too. I'm very curious what those are. All right. We're still in like the first 1/8 of the slide. All right. Let's see what else there is. You have to go host a thing, so you have to go have a good rest of the stream. We will, and good luck with your thing. Hope hosting isn't too stressful for you. But thanks for stopping by. You're in like the, I feel like maybe 5 times like the first person in stream. Like that's your running record so far, I feel like. But yeah, have a good rest of your day. Ooh. Weird little thing. Which reminds me, and I will put this on Discord, but I actually won't be streaming. Oh, you tend to be early for everything? Yep, same. I can commiserate with that. You're procrastinating on working stuff for tomorrow? Me too. Streaming gives such a great excuse, like, oh, I have to go do this. It's definitely very important. I feel like that's the best thing about that I have found about streaming is it's a hobby that has a very, what feels like a very important, like, oh, I have to do this. So it keeps me going. But this all reminds me, I will be not streaming. I will not be streaming next Sunday. I will be out of town. What is this little croissant? You're gonna do a co-work stream later, but you'll see. Yeah. If you're on when I'm trying to work, I might join. Yeah, I don't know what this is. It is a croissant. It's a microscopic croissant. I'm definitely gonna take a picture of it. I don't know why my phone. I really have to spring for the fancy version of this app to let me adjust the brightness. It also could be a super secret Ferengi ship. That's true. It also gives like Ferengi ship vibes. Zooming away, and you gotta work on photosynthesis and cellular respiration notes for literally tomorrow. I mean, I work on all my class stuff basically the day before. You don't have to be more than like a couple pages ahead of the students. It's fine. And that's what I have to work on. I have to work on my next lecture on, oh, what is it even? Like RNA, or transcription regulation or something like that. I lost the croissant. I'm distressed by my loss of croissant. You can't wait to get to teach. You can't wait to get to teaching about DNA. My favorite subject. I mean, I'm literally designed a course around basically DNA, gene transcription, and all of that, just so I can talk about it. And you've already been showing them things on CRISPR. Nice. Yeah. DNA is pretty cool. Being a French bread product, it didn't last long. Yeah, seriously though, what did I do? How did I mess up so bad? Where is my croissant? What is this stuff? Oh, wasn't it in the right plane? That's probably why. I bet it was just like little rocks or something like that. Again, we still are in like the first 1/8 of this slide, so there might be more croissants to discover. Ecology is your top tier favorite unit. I appreciate it. I have to say it's not my favorite, but it definitely gets a lot of discussions going. Students love to talk about ecology stuff, as they should, because it's an interesting topic and has a lot of, impact on their lives. It's easily like understandable as like, this is a bad thing if we do it this way. This is a good thing if we do it this way. What is this? Not those little dancing circles off to the side. They're great, but what is this? Ah, Ambus, welcome. Just doing some microscopy stuff. Got some new samples already yielding great results. You were out with COVID for all of the DNA last year. That's awful. That is the worst. I mean, I hope you don't have lingering symptoms from COVID, but it's kind of the worst that you missed it. Yeah, what is this conglomeration of stuff? We're starting to sound like we're customers at a cafe angry about not getting our complete order. Well, this time it is kind of my fault because I'm the one that zoomed in so far. But what is this blobby thing? Is this a cell? Also, those little circles are still dancing in the corner, and I love them. Embess, I hope your Sunday is going well. What are you? Okay, I should stop touching it. Oh, it's moving. Look at the little within the inner circle, there's like little dark bits that are moving around. It's definitely a cell. Blob thing at least has a cell wall, question mark. Some kind of reproductive structure, like a little tiny egg? Cyst? Yeah, it's hard to tell. I don't know what this is. I mean, who knows what type of, reproductive cell? Like, I'm wondering if that, see how there's like a darker, or like another circle with, so outer circle, slightly smaller circle, but then in the dead center, there's a little tiny something. I wonder, a statoblast from a bryozoa. Let me look those words up. I know what a bryozoa is, but I don't know what a statoblast is. Oh? Well. It's like we're in the right geometry, but these are a little big, and I think, well, bryozoa can be freshwater, so. Well, through the process of elimination, we know it's not a horse. True. We can just kind of get that out of the way. That's very clear. Well, this thing is kind of cool. I wish it would do something, though. Although by observing it, I think we're gonna probably hurt it. I just kind of want to watch it. You know, I'm gonna stay here for a little bit because I'm curious if there's any like broad movements that we're maybe not noticing. So I'll take the video from stream later and just kind of let it like fast forward it and kind of see if there's anything more noticeable. So Bryozoa preserve themselves in harsh environments to try to form a new colony. I don't think it's the primary mode of reproduction. You don't know. And it's starting to feel like it's looking back at us. Yeah, a little bit. Oh. Mark 5 Sneakers, thank you for following. As I'm just sitting here quietly watching something happen. Everyone else can see the little thingies moving around, right? So that's a bit fast for what could it be? I'm waiting to see if like there's gonna be some like DNA condensation or something like that. Yeah, everyone can see the thingies. They're moving around a lot. Is it organizing itself? Also, now the whole thing is starting to twitch and move. I think something's, yeah. There's a hint of structure inside of it, like that circle that I mentioned, like inner circle, right in the dead center. It's a rave. Does my rave button work? Yeah, there we go. Rave. Yeah. So, okay, probably a eukaryote. Eukaryotes love to move stuff inside of them around on like microtubules. It could always be something like that. Just little vesicles moving around, which is actually kind of important, kind of cool that we can see it, if that's what's going on. I was gonna say it's a little fast for like chromosomes to be moving around. Not that chromosomes don't move at this. I don't know. It's a little fast for yeah, that. Some type of fish egg. Again, this was okay. I would love it if this was a fish, only because remember this is from the bottom part of a flower pot that has been sitting on our porch for a week. If a fish egg got in there, I would be very pleased, but I don't think so. This is also at 40 times 16 times like 10 magnification. That's an itty bitty fish. Never say never. That's true. That's true. Okay, this thing is interesting, but I just don't know what's going on here, so I'm gonna leave it. We can I'll take a microscope shot, and maybe we'll come to some like inspiration later. Pollen, probably not. So all the pollen that I've seen has a pretty like I was about to say thick for this scale outer shell. You can really normally see it quite well, so I don't think it's pollen, but yeah. Ooh, why is this stuff moving? Well, we have all sorts of little things like stuck in this little blobby mess. And is this filament right here? Oh, I thought it was going to lead us to another ciliate. Not really. And we still have our little dancing friends. They're having a little party right here, and they don't want to leave. Look at them go. All right. Let's zoom out. I'm at very... Wait, now that I zoom back out, it looks like it changed. Doesn't it? I wish I had a better... What? I just... Like, are there two little circle things inside of it now? I wish I had a better objective. Slash, I wish I had a better microscope. I'm so curious about that thing. But we're going to leave. We're going to leave it. We have to say goodbye. All right. I have to zoom out, I think. Ooh, actually, I have to zoom out after I investigate what these are now. That is a broken diatom. What was this over here? These little filaments are definitely something. I just don't know what they are. Super descriptive. Only the best descriptions. Yeah, okay. Zoom out. We're gonna zoom out. It's easier to see things. Again, I think we've actually gone back in terms of like how much of this slide we've seen. Like we've actually seen less of it now. Googling ciliate cyst gives you something pretty close. Well, you've got my attention with that. And I'm going to continue to not fix this microscope thing right here. Ciliate cyst. Fish egg would be way larger. Oh, absolutely. Ciliate cyst does give something very similar. Huh. Yeah, you can definitely see them with the naked eye. Yeah. We were just kind of joking. At least I was joking. Yeah, it's way too tiny. Yeah, there are a few that look similar as I'm kind of like zooming through here. Not exactly the same though. At that scale though, we can probably just say, oh yeah, it's a ciliate, and probably be right. Yeah, I know you were joking. All right. So, what else do we have here? Oh, hey, look at these. A new delicacy of the ultra-rich micro caviar. See, I am convinced that with a good marketing team, you could market so much like normal things with enough science-y language to make it super like attractive to rich people. Yeah, what's this? Yeah, are these hyphae from a fungus? So they're coming off of something that is very big. I actually might need to zoom out a little bit more. This is still the flower pot. Still talking about that flower pot. You don't even need scientific language. Just look at the lobster. That's true. Oh, look. Look who has attached themselves. Okay. Let's see. Ah, welcome. Yeah, you're welcome. It was fun. I think that was Alt Miss Frizzle's idea. Yeah, welcome to the stream. Thanks for chatting. Oh, and then you would then you would end up with those useless home gym equipment whose effects somehow are scientifically proven. Oh, there you are. Look at him. Oh, he's eaten. He's trying to get these ciliates. He moves so fast. It really is like his eating animation is missing like a frame in the center. Come on, you can do it. It's right it's right there. Come on. He needs to wait like 2 seconds. Biology is crazy. Yeah, no, it's yeah. Well, yeah, welcome to the microscope stream. It's we go around, we find new little friends and watch them live their lives. At least we try to. Come on. Meanwhile, I have to like I'm actively trying to keep up with like which plane he's in. Did he get it? No, it's even worse because those little ciliates, I think those are ciliates. Yeah, they're getting farther away from him. Oh, he's trying. Oh, oh, look, a little bit of algae's right below him too. Yeah, he's pushing it away. What are you doing? Oh, David, I love this already. Dirty water with extra tardigrade protein. Good grade, better grade, best grade, tardigrade, which means top quality. And the power of E. coli will help you lose. Ooh, yeah, will help you lose weight. Fast, dirty water. Order now. Did he get it? No. He might be not trying to get those. Might be going for bacteria. That's very true. Yeah, you're right. Uh, come on, get over here. Yeah, you're right. So he might be going for something we can't quite see. And those might just be sort of attached to something nearby. It's tough. I am gonna look and see though. He's attached to something that's got a little bit of movement here. Oh, oh, hello. Oh, so we're definitely looking at the surface of something. I know I zoomed in, but I really need to zoom out so we can check out what this is. Yeah, he's a like what's a word that you can use for something that just eats a prokaryote? Oh, he's going at with his little propellers. Look how fast. So for anyone who isn't catching what's going on, this little trumpet-shaped thing has little propellers on the sides of its mouth, which are kind of fuzzy. They're going really fast right now, and so all the things flying by are because of the little vortex he's making. Prokaryotroph, right? That's gonna be a word. That's gonna be about right. Yeah. See now he kind of he's just kind of moving things. Now that he's stopped, though, I can finally like zoom in a tiny bit more. Let's see if we can kind of. I can't really make out much detail there. Just see his insides moving around. Yeah. I feel like my like. I don't even know what it'd be. I don't even know what like subscriber goal it would be, but like get a new microscope. It'd be pretty high. Oh, he's really raking in some stuff. This wasn't here before. He's so fast. Like, the stuff kind of going straight through, like diagonal through the frame, is just like whizzing by. Wow. Stir that syrup. Okay. I'm going to leave him alone to kind of finish his meal because I'm going to zoom way out. I don't even know what we're looking at this point. We're looking at something. There he is. this big thing, and then there was some hyphae or something. Oh, oh, is this a seed? Okay, this is too big for pollen, but this could be a seed or something. Or maybe like a very, very small plant. Not duckweed, but something, I'm thinking a little bit like duckweed. What is the highest? Oops, hold on. Let me zoom out a little bit more. What is the highest magnification I can go on this microscope? So what we were just on to zoom in all the way was 40x times 16x. That gets us to what's at the eyepiece. And then I have a digital zoom of who knows how much, probably another like 2 to like 10x digital zoom. So, pretty good. But the problem is, so the problem in microscopy is like when we're zoomed out like this on this particular microscope, there's something called the field of view. So it's like up and down. So on the screen, it's towards and into the screen. Right now at this view, we can see a big chunk of it in clear focus. But as I zoom in, especially to that 40x, it goes from like a field of view of this to like this. And so you only get the tiniest sliver of something that's actually in focus. And no amount of digital zoom helps with that. Yeah, I have a 16x eyepiece. I honestly kind of wish I also had like a 10 or something, because every once in a while we see something that would be nice to actually like look from far away. Oh, thank you, Embus, for contributing to the pack of cards challenge. I'm just trying out the challenges thing, and I have a bunch of packs of cards to open, so why not? You want a 10x? I feel like 10x is actually more common, maybe. Okay, I'm going. We're zooming in. We're zooming back in, by the way. We might go all the way in, actually. I want to look at what, I don't know if they're hyphae or if they're growing roots or something, but we'll take a look at them. Oh, there's another ciliate chilling right here. It's like our friend we just saw, actually, except that his mouth is closed. You only have a 25x at school. 25x is still pretty good, and honestly is probably a little bit more high schooler proof, because like the 40, I can very, very easily accidentally smash the 40 right into the slide. I have definitely done it. I've definitely probably shortened the lifespan of it. 400x would be perfect magnification for what you want, but you get either 250 or 1000. Yeah. So wait, would a 10 help you with that? You looked up fungal spore to see what it is, but you don't think so. But you think the French bread product is the one that we couldn't find, right? Yeah, we found a thing that looked like a croissant, but then I lost it. So I think this is another one of these stalked ciliates. I think it doesn't have its mouth open. In fact, oh. I don't know if that was a good thing or if that was it moving one of its like vacuoles or something like that. Anyways, I think you can kind of see where the like folded cilia of its mouth are, kind of in the upper, what is that, left-hand corner. TEDx and some depression slides would make you happy, lady. Yeah. You should check out, I always check out university surpluses near you, or even state surpluses, because they soak up university and school materials. I've found a lot of the not equipment, but because this microscope I did get from somebody, but everything else I find usually at surplus stores. Okay, so these hyphae, which are not hyphae, maybe are sort of more a root sort of deal going on here. I've got so many little little guys. There's another one. This sample is just chock full of them. Here they are. Here they are. Yeah, so what are we looking at then? You work with the Sam Houston Medical School. Nice, yeah. Um, labs shut down all the time. Like even private companies will just donate their stuff. I actually can get a lot of good scientific like accoutrement from like what's like a craft store. It's like a thrift craft store where they just buy a bunch of like random junk and then sell it as like random craft bits. Okay, so is this a root tip? Root tip. Because according to Micro, it doesn't look like a fungal spore. Then again, whatever it is, I think it's something planty, but whatever it is, it could be rotting, and this could still be like a hyphae. Hyphae? Hyphae? Okay. I gotta look this up. Oh, the big thing. Oh, okay. Root tip. Root tip. Def hyphae? Okay. Hyphae. Microscope. Yeah, I've seen hyphae before, but not when they have like this clubbed ending. That's what I'm a little bit confused by. I remember the cells being a little bit, I don't know, they just looked a little bit different. They looked ready to like sporulate or something like that. Yeah, I'm not a. Oh, unless this is a sporangium. Okay. Yeah, I could get on the hyphae train. Ooh, yeah, wait. Okay. Root tip microscope. Oh, yeah, absolutely not a root tip. Why you should have, all of you should have just shut me down immediately. Here, let's look at this. Yeah, no, all misfits. I don't think so. I mean, here, let's compare and contrast. These are stained. Sorry, I'm covering chat up. You get covered. Oops. Yeah, the organization of the cells. And, right, because root tips are multiple cells growing in parallel lines with each other. And they have a protective cap. It's all coming back to me now. Granted, these are like plants, like organized plants, and this could be something a little bit less macroscopic. Yeah, no, these are hyphae. These are hyphae. They're really good at showing mitosis. Absolutely. Something I did want to try one day. I just don't know how to prepare root tips for slides. So that's a activity I have to do eventually. Let's go back to the microscope. Yeah, hyphae. Hyphae. Ooh, ouch. Okay. Hyphae plus friends. So it looks like these are coming off. These are just coming off of some planty bit. And all these other organisms are sort of attracted to this for whatever reason. Ooh. Oh, is this a big one? You know, we just keep finding these guys. And it looks like, I wonder if that's its meal, its most recent meals. You can see its little like bottom propellers right there, undulating. It looks like it's taking a break. What kind of plant was in the pot? See, so it was, we have been calling what it was a Mexican sunflower. Great plant if you want to attract lots of hummingbirds. I don't know if that's actually what it's called. But the thing is, like, it's also been like just on the ground, and we have lots of other plants, and there's like so many other random leaves and petals kind of falling into it. Nothing that should be giving off seeds that are like tiny like this. So I think this is just something else that probably fell in it. I mean, because we have like, it's where it is also like covered up sort of by like tree cover. So any number of things could have just fallen straight in. So yeah. Is my mic low? I feel like it's showing me that it's low. I'm just popping it up a little bit. There we go. Not a lot of meat on a hummingbird. They're hard to catch. I literally was telling my cat that this morning because she was obsessed with the hummingbirds. But I don't think, yeah, they're not good eating, I assume. Is this yet another one of our little attachment friends? Oh, I don't see his attachment point. Looks pretty similar. I don't know if people remember or were here, but I would love to catch a Stentor. That's on my microscopy goals, my vision board. All right. Yet again, distracted. Why is it so yellow? All right, I'm gonna zoom over a little bit to find another little neighborhood. Got a bit of rotting leaf there. See, now that I said I want to see a Stentor, I'm seeing Stentors everywhere. They're the little trumpet-shaped guys. They also kind of attach to things, but they don't... I don't think that was Stentor, actually. Wait, have these been Stentor the whole time? I thought they were more trumpet-shaped, less ball and chain-shaped. Stentor. Stentor ceruleus. They also have a fairly distinct color. Yeah. No, I don't think that was Stentor. They're not dissimilar. Yeah, okay. Sorry, going fast. So there's actually multiple of this weird like thing. It's the same thing we saw before. You know, it has to be a seed, right? It might just be a little tiny seed of something. Because you could almost imagine that that center part is the the plant embryo, and just the rest on the outside is just like the seed coat and the endosperm, I think it's called. Yeah, oh yeah, look at that. Could be mycorrhizal fungi growing with the flowers, potentially. I think they're definitely, these are definitely seeds. So that other one was just a little bit more deformed. What the heck? So we have purple colored trumpet shaped flowers. What is that? What are those? What are those called? What are those called? I'm blanking on the name. Petunia? Petunia seeds. Show me. Oh, they're little tiny. Yeah, so this pot is sitting underneath a petunia. But I'm actually not finding a good picture of just petunia seeds. Not a zoomed in one. Oh, wait. I'm not convinced. Okay, I don't think these are petunia seeds. No, they are not. Absolutely not. There's something smaller. I don't know what they are. Clearly clams. Yeah, they're my micro clams. All right. I think this has been a wonderful sample, but I think it's about time to at least check out the pond water. I mean, it's I don't know what's in it, but it's fresh. It is from like I scooped it like 5 hours ago. But look at all these ciliates. Yeah, so I might kind of keep this sample around and just on my desk and see what happens to it. See what grows. See what dies. Stentor? No. I want my Stentor. Clam seeds that grow into grown-up clams. Yeah, exactly. We are several hours inland from the sea, so these clam seeds, they actually fly on the wind into the ocean before they turn into full-size clams. Yeah, baby clams are just little tiny, almost microscopic clams, aren't they? Baby clam? What will come up when I say that? When I search that? No. That's not gonna give me the image. Immature clam. Whatever. Yeah. I'm gonna switch this out. We're gonna go to pond water. If it's not super interesting, we'll leave immediately. Here, let me cover this up and then show the camera exactly what we're dealing with here in terms of like this sample we've been looking at. I don't know if you can see it, but it's like disconcertingly brown because it's like tea, but made from things that fall on my porch. All right. I don't have. We shall ponder the water. Yeah. I need to go get something to clean this up, so I think I'll just put a bunch, like too much pond water, in a larger sample dish. I will let everyone watch that while I go clean up some of the slides. So I also noticed some like goopy things in the bottom of this that kind of settled. I'll try to grab a variety of things. So this will also be kind of useful because it will kind of settle as we're sitting here and talking. Grab some sediment, grab some detritus. There we go. And if I'm off cleaning something, I won't be shaking the slide. You love catching tiny clams at the beach. We call them sand fiddlers. Huh. I think they're coquina clams or something like that. You feel like a kid again, chasing after them in the sand. Hello, science. Welcome. Yeah, obviously go check out science. I don't think there's anyone here who doesn't follow the team over there. But just in case, do it. Yes. Hello. You must be streaming later today, if not imminently, don't you? I feel like you normally start streaming around now. Uh-oh. That is that's a great reminder. I have not opened my water. I have not seen the new printer. Oh, resin printer. Oh, I'm very jealous. That's like, hopefully a future purpose. Very soon, but you want to come say hi and hang out. Well, thank you very much for dropping in. Yeah, got back from camping, so obviously I grabbed some pond water samples before I left. Yeah, you knew I'd be jealous. Yeah, see, that would have been. I'm taking a little bit of break with the like microorganism mazes, but I really am. Oh, hello. Is that a little Daphne I just saw? Um, I do really want to like resin print the next like set of mazes because I think that will be better with our little microscopic friends. Um, what are we looking at? We are looking at a sample of pond water from a freshwater pond that actually recently underwent a like severe infestation of cyanobacteria. It was actually dangerous to be in just a month ago. Um, also, hello, Cliff. Welcome. Hello. Oh, and I didn't. If I didn't say already, Hoba, welcome. I don't think. Well, it says you're first time chatter, so I assume you're new here. Yeah, so we that our fast new little friend was a Daphnia that just swam by. At least I'm pretty sure it was a Daphnia. They are a type of freshwater crustacean. Seeing some also seeing some nematodes here. What's the resolution on in magnification, if you don't mind? I always forget what my objectives are. So we are at 4 times 16, and then whatever tiny amount of magnification my phone camera is also giving us. So you know, 4 times 16. I'm definitely not writing that down so I can do the quick math because I'm bad at mental math. So that's 64x magnification. The ultimate goal of the mazes should be to add a chemoattractant. Could not agree more. Absolutely. However, finding the right organism with the right chemoattractant has been difficult in my like side research that I, am doing off stream. So please, if you've got ideas about chemoattractants and microorganisms that are attracted to them, put them on the Discord. That would help me out a ton. Wait, what am I doing? I said I was gonna just set this up and then let people watch it while I go clean off something quick. I do need to go do that. Okay, let me find something quick. I'm just too addicted to like zooming around and whoa, hello. And a Daphnia. Look at that. I think that Daphnia was had a clutch of maybe. I feel like the dark spot is a clutch of eggs. I don't know what this is. Just some kind of giant worm. Very, very long boy. You know, actually, I feel like we have seen one of these guys on stream before, like a long time ago. And also, hello. Yeah, it's some kind of annelid. Look at, got another little friend right here hiding out from the giant scary worm. Okay, I'm going to zoom out a little so you can watch that. I'm going to turn off my camera and I'm going to go get some stuff to clean up. So, be right back. All right, back already. Perfect. Yeah, look at this little guy. Ooh, also there's lots of little things in here. That looks like it might be a nematode. Yeah, absolutely is. A little slow. Wow. Is it gross that wait, how do I word this? It feels kind of disgusting that what I thought was a dirty, infested pool of tepid water, of fetid water on our porch looks actually more clean than from this freshwater lake that a bunch of children were swimming in when I was there. Maybe I should be surprised? Yeah, it is a super busy sample. I would love to see that Daphnia again, or a Daphnia. Because if we can get a Daphnia under the slide or cover slip, that would be nice to see. Oh, hello. What's this here? Couple things. I feel like I'm seeing movement. Oh, I am seeing movement. Is that a worm? Or something? Do you cover slip all my samples? No. I only do that when I want to use my 40x. Just because that's the only way I can do it. Also, it prevents this shaking because it squishes down the plane of everything so much. The only issue is that if I don't get anything interesting, then we're kind of out of luck. Your anti-cover slip. I do for certain things. Also, it comes back to the problem of like field of view and all that stuff. Like, how do I see all the things I want to see? I feel like this is some sort of insect larva. Oh. Hello. I was just about to say, I feel like this is some sort of insect larva that's hiding out in like a little house. Yeah. Yeah, you know what this is? I know what this is too. What are these called? This is a very small one. You need to remember what it's called in English. Yeah, let me look up. I need to insect larva. They make like little houses, little house in water. That's what I'm searching. Insect larva, little house in water. Yeah, this is too big, but it reminds me of a caddisfly. Sorry, a caddisfly is too big. This is very small, because caddisflies make little homes out of pebbles and bits of plants. But this is way too small. Unless they do it at smaller stages of their life cycle. I don't actually know that. And we scared him off. He hasn't come back out. So, whoops. Let's zoom back out. What are you? There's also been that thing. I can't quite tell what that is. Okay. So, elsewhere on here, I need to move it. There was also something very big and very weird. I don't think it's going to show up too well, but I'm going to attempt to move it, and we'll double check that. Something big and squishy. Yes, here we go. So I don't know what this is. It looks like a bit of plant, but I wasn't sure if it was something interesting. It was at the bottom. We're not going to be able to see very much for that. Okay. Oh, I saw a copepod. All right, we've got a very, very lively set here. Now, how to do this? See, because also if we got some things under the cover slip, they wouldn't move as much, and we could watch them better. The only problem with this sample is it's not a lot of squishy stuff. There's a lot of bits of rock. By that I mean sand, which could be good. It kind of keeps things from being squished down. It keeps everything within a plane. But I really want to make sure we catch something interesting. Oh, hello. What are you? Let's zoom in on this. If it would please get out of the way of that stuff. Yeah, until told otherwise, another annelid. Just way smaller. Whoa, hello? There is something on this one. I just saw Daphnia go by. There you are. Daphnia are a ton of fun, so I really was hoping to get some. I'm glad I did. But that's going to be hard to grab out. This culture too, I will let culture. This sample, I will let sit on my my desk for the next week. Ooh, although, yeah, because I won't be streaming next Sunday. And this Wednesday, I was actually thinking about going back to that programming problem that we were tackling a bit ago. You know, converting images into DNA sequences. I was thinking about getting back to that. So I don't know if this sample will still be good by the time we get back to a microscope stream. All right, I am fairly well convinced that I love this sample. I'll put some on a slide and we'll see what happens. If it's bad, we'll just come back to this. Actually, what am I doing? You can go ahead and watch this while I prepare that. And if it's boring, we can easily come back to this or go back to the dirty water. Or I still have a bunch of things sitting on my desk. The tardigrade culture unfortunately got away from me and is no longer what I would call good. There's still some tardigrades in there, but it's kind of been overtaken by algae, a lot of diatoms, which are great, but not exactly what I was looking for. And so I wouldn't say we're also like waiting, but I did send in my DNA for sequencing. So hopefully that'll come, sooner rather than later, but that's still going to be potentially like months away. That's going to be fun to look at. Got away from me and it's no longer what I would call good, relatable content. Yeah? I'm nothing if not relatable. How dare you, I don't know, suggest otherwise? You're not suggesting otherwise, I think. So, I mean, I don't think of the microscope samples as like my pets, but they feel like, I feel like my feelings towards them are almost like neglected plants. Like they're not my favorite things, and I do try to take care of them, but I also do neglect them a little bit. So that's how I feel about them. Oh, I need more water. Oh, we could also look at a dead wasp. The wasps that came out of the caterpillar, I do have one or two that I kept before I released the rest out. So much stuff. If it were, I gotta change these lights. She-Hulk colors. If it were any other week, I would stream longer, but I have so much work to do tonight for class this week. I really need to stick to my, done by 4 schedule. All right. Who? Wait. Oh my gosh. Could it be? My dream came true. Stentor. I found a Stentor. This is so exciting. I just wished for this to happen. Oh my gosh. Look at it. You can tell it is by the way it is. Yeah, no, so Stentor, dreams do come true. Best Sunday ever. Wow. Come on, stupid microscope slide. Look at it. So they... Let's see. Wait, I love you for that reference? Did I just make a reference? I feel like I make too many references to know the difference between when I come up with something myself or I'm just accidentally referencing something. So you'll have to repeat it to me. So Stentor, super, super cool. They have this like trumpet-shaped body, like old style trumpet. Oh, yeah. Yeah. Nietzsche, exactly. See, I didn't even realize that I said that, which is frightening. But that video is definitely like stuck deep in my brain. Oh, it almost got that little guy. Okay. So long trumpet-shaped body. They have a mouth up at the front. These guys are fantastic. They are huge for a single-celled organism. Go down. They also have axes, body axes. They have a I think they have a left and right, but they have a head, they have a tail, they have a dorsal and ventral, I think. It's kind of because their mouth, you can't really, maybe you can kind of tell. It's shaped a little bit like a curly C. So the reason why that's kind of weird is because like we have body plans, but we have many, many cells to help us differentiate that, right? As soon as you go from 1 cell to 2 cells during cell division, well, suddenly you could start setting up a body plan because you went from nothing to left and then right. You can kind of tell the way its mouth is shaped. Oh. That was exciting. It's like an action movie. Anyways, he keeps doing that. These guys we were talking about a week ago or something like that because they have a crazy amount of nuclei inside of them, something like 10,000, and it was sort of unclear at the time and in retrospect, but it seems like each of those nuclei has the potential to be extremely polyploid. So like greater than 100 ploid, it seemed like. I may have been misreading it at the time because I thought they were saying it was like 1,000 to million ploid, but that might be like 1,000 ploid times 1,000 nuclei. Anyways, I've been trying to like get this to focus, but they have little beads inside of them. Those are each of their nuclei. Their mouth, when they get overused or damaged, or if they have like a particularly large meal, they will eat their own mouths and they will grow a completely new one. If I were somehow able to poke this thing, rip it in half, both halves would very, very likely become 2 new Stentor. Their the cytoplasm inside of them, the goop inside of their cells, has this property that becomes extremely viscous when it's exposed to the outside. So they're very, very damage resistant. Like you poke them and any other, like if it was a human cell, you poke a human cell and they just kind of burst. All their stuff flies out. These guys, basically heal up in a certain way very, very quickly, and then they're able to survive. Anyways, welcome to Stentor Facts. I don't know what species this is. I think the not normal species. The famous one is Stentor ceruleus. Ceruleus comes from the cerulean color. Because of my camera setup, it's very hard to tell what this is color-wise. I'm going to attempt to use an overhead light, which sometimes works, but I think it's too small. So this is going to go dark for a second. Oh, yeah, look at that. Yeah. It's a little bit of a cerulean greenish color. Microscope picture. How do they contract and expand? That's a good question. So they probably have some sort of contractile fiber system inside of them. I just don't know exactly how. So let's see. Ciliate, contractile. They also might have a contractile vacuole. Let's see. I guess I should say Stentor. Stentor contractile vacuole. Yes. So they have a contractile vacuole. It's a sac basically of water that is of a different oh, you say contractile vacuole is for peeing. I thought they also used it to move, though. Is that not true? I guess they just say discharge it from their vacuole. Well, then I don't know. I don't know how they're moving. Movement. I'm just Googling at this point. Movement. I mean, because they're gonna have architectural proteins inside of them, like actins and microtubules of some kind, probably. But they probably don't move like we move. Let's see. But who knows? Whoa, something big just went by too. All right, this Stentor has been the highlight of today for me, but I think I'm gonna move on. And wait, what are you? Whoa, I do not know what that was. So the Stentor is doing something. Oh, he's just moving. He decided it was time to go. Whoa, that was interesting. So that's cool. He's putting on a little show for us. So we just saw that they too can attach and detach to things. They just don't have as long, or at least this species doesn't have as long of a tail as the other one that we were looking at earlier on. Which wasn't a Stentor, but it was another ciliate. Okay, I was going to try and zoom in a little bit more with a different objective, but if it's moving around this much, I don't know if I'll be able to. Oh, you found it. They use myonemes. Myo probably meaning muscle. Thank you. A contractile structure found in some eukaryotic single-cell organisms. Also, I see that they're leaving. Oh, is it oh, no, that doesn't seem like a good source. Hold on, gotta at least make sure I follow along a little bit. I do feel like I missed my chance to zoom in more. Have to head to lab. Sounds good. Yeah, we'll be finishing up fairly soon. But thank you for stopping in, Brian. Okay, come on. I'm trying to learn about your muscles. Okay, so they are made of bundles of microfilaments twisted up into microstrands. The organelle is dependent on calcium concentration. Interesting, interesting. All right, so it kind of is a little bit like muscle. Just a bit. I'm not actually finding a lot of good information on it. I know I just kind of lost it. Okay, so let's see. Flagellum. Nope, not flagellum. Thank you for this, but I think I'm going to like look this up later because I just lost him. I want to keep looking. Did I say organellize? Did I? Maybe. That sounds like something I could have said. Lost him. Oh, no, I didn't. All right. Well, this is great, but I'm going to go somewhere else and look at the other things we have. Like this crazy thing. Wow. Next time I'm hemming and hawing about using a cover slip, just tell me to do it. I don't know why I was afraid that I wasn't going to see anything. Like I was going to miss all of these microscopic things. Hello? Are you something? Maybe. Oh, yeah. What do you think? Diatom? You found a diagram of the structure? Link it and I'll pull it up. And that little friend, which might be another ciliate that likes to attach, if that little tail is something. Baby Shark is playing too loudly. You want me to remind you not to use it? No. No. Oh, in the Discord. Awesome. Thank you very much. I will open that up. No, I'm team cover slip, clearly. Oh, clips. Oh my gosh. Oh. Okay. Okay. I'm getting information here. Mainstream. All right. Ooh, that's tough to see. Oh yeah, the gullet. Oral pouch and gullet. Yeah, so so the myoneem I think is the full structure, and it has microtubules going in what I think are multiple directions. And so those microtubules, by some mechanism, are going to slide past each other, which is not dissimilar from how muscles work. Very similar idea. This doesn't, I see you might have said something. Source, thank you. Let's check this out. Ooh, regeneration in Stentor ceruleus. Scroll. This was in B. Oral pouch. Ultrastructural organization of ciliary rolls. Parallel arrays of microtubules known as KM fibers. There's a contractile fiber bundle composed of centrin-like EF and calcium binding proteins. All right, if that's kind of all it says, well, yeah. All right, it's a little late to read the whole thing, but thank you for this. Everyone who's interested should just go check it out on the Discord. Because it looks like it has a lot of great information about Stentor, just generally. So that's awesome. I'm definitely going to go read it when I have some time. And by that I mean when I'm supposed to be working on my class tonight, but get bored / need a break. Read about some Stentor. Hello. This looks like the cast-off shell of a, what do you call it? Copepod. Yeah, thanks, Micro. I do appreciate when everyone doing the real-time Googling for me. It is hard to Google and microscope at the same time. A copepod ghost. Very spooky. I thought so. Is this another one? Thank you. Cheers to Micro for doing the work. I think this is another Stentor. If it would just go ahead and scooch out a little bit, we could tell. But I think I'm going to center this time. Not make the same mistake as before. Whoa! Oh my goodness. Welcome, Volcano Doc and 16 friends. Thank you for the raid. Hi, everybody. Now I got to zoom back in. Welcome. By way of introduction, my name is Dr. N. We are doing some microscopy today, and I've been very excited to find this little friend, which is called a Stentor ceruleus, or Stentor, maybe not ceruleus. It is a fun little ciliate who has a bunch of crazy biology related to him. And I was just about to zoom in, but I will just take a quick break to say hello to, Volcano Doc, thank you. Dracula, Passing Wind. Yeah, welcome everybody. I hope you like, so I assume you're talking about volcanoes. So now we're going to go a completely different scale. We're talking about the little things that live in the, freshwater near you. Ah, yeah, look at this. I'll zoom into his little mouth. Ooh. Oh, and there's some nice little algae there. I wonder if he's going to try and eat it up. And a diatom. And some other... Oh, there's a spirally sort of looking thing. So you were talking about EQ today, but it's all geology. Okay, Nadak. Oh, you're assuming you're going to grind rocks today. Grinding rocks. Yeah, diatom. Well, this diatom might be about to be consumed, but there's a diatom right there. Yeah. Oh, this Stentor has a lot of little fun things kind of caught up in its little web. Amoeba or nostoc. Yeah. So this really, or is that nostoc? Because hold on. Again, because of the way it is. Nostoc. Let me see. I don't know what I'm saying, but I would have guessed that was nostoc. Let me check out Anabaena. Ooh, that's hard to tell. Oh, your regular job, you make microscope slides of rocks. Hate to rain and dash, but you have to go work. Yeah, no problem. I feel you. I do the same thing. But thank you for the raid. And welcome also. Ooh, this, come on, Stentor. So this has given us a really great view of its mouth right there. So you can see, what was that called? It's like pouch, like oral pouch. Gotta zoom back up. Actually, this well-timed, image you sent me. Whoa. Uh, Thank you for the follow, Passing Wind. Yeah, so we're looking at its oral pouch right there. So that and do you see that sort of almost invisible bubble right there? That is its contractile vacuole, not vesicle, vacuole. Yeah, it does look like the head on a fingerprint. So I think that's actually, um, if I remember right from other images I've seen, I think that is actually related to its contractile abilities because its mouth can sort of snap, not shut because it has a geometry that's hard to describe, but it kind of has this C shape that it can kind of close a little bit. We're getting a really nice view of its cilia though. Let's see if we can find its nuclei. It's got enough of them. Come on, come on, come on. Microscope. Behave. Oh, actually, you can see also its myonemes, or whatever you want to call them. The parallel fibers. If I kind of zoom, change the focus just a little bit, you can see fibers that are going sort of parallel up and down. Ooh, especially there, and especially near its holdfast down here at the bottom. Actually, let's find it. Oh, it's stuck way back here. Myoneme is the word I learned. Myoneme is the word I learned as well today. We've had some quick Googlers here who have really helped define what the structures are in this particular organism. So like I was saying before the raid, this organism is really interesting because it has many, many, many nuclei, but it's a single-celled organism. It also has many, many more copies of its genome. So I was actually kind of hoping to get just the right focal plane to see if we can see its string of beads nuclei. The scale is probably just not what I'm expecting. But I think it's on the other side of it from its contractile vacuole. Not quite. Oh, wait, I think I see a cell that we had seen outside of its body and it's now inside. Oh, no, it's right there. Yeah, so what it's attempting to do, it's full of genes. It is full of genes. We actually only recently, I think within the past 5 or 6 years, maybe longer by now, fully sequenced its entire genome. It is super fun. It is like it has all these nuclei, right? But its genome is extremely compact. It has introns just like other eukaryotes, but its introns are something like 15 bases long. Oh, where is it going? I think maybe it's not leaving. 15 bases long is basically the physiological limit. All right, this one's swimming away. She decided she's done with this area. Any fewer bases for your introns, for any molecular biology nerds, any fewer, and you basically cannot have your RNA loop around to make the lariat required. All right, she left. Oh, no, she's still there. Love to play with genetics, but you got reading and DNA is expensive. Reading and writing DNA is expensive. Yeah, well, if you're interested in, I just sent in a blood sample for myself to get, fully genome sequenced. And I hope to have the results in a month or two. So we'll be checking out my genes in the future. But the DNA sequencing technology is getting to the point where it's affordable for, you know, normal people. The reading bit does take a bit of knowledge, though. You can pay for services to get that done for you, though. It's interesting that it must also mean that they're using them for something. Yeah, so, introns are the ancient ancestors of DNA invaders, and eukaryotes have co-opted them to create a huge amount of variability. Is that an amoeba? A huge amount of variability come on, finish the thought. Huge amount of variability in our genomes in order to create all sorts of different protein products. So they're probably still serving the same purpose, but what they're maybe not doing. Oh, that is an amoeba. What they're maybe not doing is doing a lot of the non-coding RNA stuff. Look at this little friend go. Wow, this is like top 10 streams right now. Top 1 microscope stream. Maybe not top 1. Top 1 was definitely the first time I found one of my tardigrades. But this is top 3 for sure. We are finding all of my top tier list of the things that I want. Can't finish a thought when you see an amoeba. Yeah, exactly. I can't finish a thought on this stream because I look somewhere else and see something cool. Come here. Come here, you. Oh, look at that. Amoebas are... It's like blebbing out. It's like pseudopods to move around. Now, is the big circular thing in the middle a vacuole, or is that its nucleus? You've seen amoeba and think of the original series of Star Trek that ate the spaceship. Oh man, it's been a long time since I've seen that. That would be a good supplemental to like a Wednesday stream or something like that, if I was able to culture amoebas correctly. Yeah, so for any of the newcomers who I see are still here, although I do a lot of just random, mostly microscope stuff on Sundays, I do try on Wednesdays to more or less stick to a theme, often sometimes closer to a theme than others, but usually about sci-fi and science or something adjacent to that. So we've talked about cloning, bringing organisms back from extinction. We've had a bunch of guests on. We talked about vampires on Wednesday. That was super fun. Yeah, we're definitely Nascent Novice will be back. I will force him to be back so we can finish? We can just keep talking about vampires because we never even got to talk about rabies. Top number 1 fear. Everyone should fear for their lives when it comes to rabies. It is a horrible disease. You wonder what the introns per gene count is compared to other eukaryotes. I think it's actually like other eukaryotes. I think there is, at least 1 intron per gene on average, but I don't quite remember. But yeah, the introns thing is super fascinating. It's like the average size was 16 and the smallest was 15. It's like 99% of their genes have a intron length of 16. Okay, I gotta take a screenshot, but this thing keeps moving. This thing's like flubber. Anyways, yeah, if you're interested in biology generally and also just talking about pseudoscientific things, that's what we do. I am what haven't we seen on stream today? We've seen Daphnia, we've seen Stentor, we've seen Nostos, we've seen an amoeba. We saw that other ciliate thing that I never actually looked up. Yes, he was a wonderful guest, and vampires do rock. Okay, I love this little amoeba, but I think it's, I'm going to zoom around a little bit more. This slide is so clearly full of, you know, so many friends. Yeah, we actually have not seen any tardigrades on this particular set of slides. So I'm not too surprised, just because at this point, tardigrades, they tend to pop out from my lichen samples, like dry samples that I wet, not from like samples that are always like literally underwater. Yes, we did see rotifers. In fact, oh, that's not a rotifer. I thought a rotifer just went on screen. Yeah, we saw some rotifers. I didn't really call it out. Yeah, so my tardigrade culture is a little neglected, because I have some tardigrades that live on my desk in a dish. It's just that they might have all died because I forgot them or something. Let's see. We saw a caddisfly. That's probably not a caddisfly. Yeah, we did see that. Yeah, no word on what that actually was. Oh, there's our ghost copepod. Very spooky. It's almost September, which means it's almost October, one of my favorite months. I'm kind of wondering if this is like the preamble to like a dry spell of microscopes things. You know, it starts getting colder in my area, things going more dormant. Maybe the samples will not dry up literally, but dry up sample wise or organism wise. We did see a tiny croissant. Not uh, oh, not to be on a Star Trek theme, but in Star Trek Discovery, they're using a tardigrade DNA bonded to a human to navigate. Yes, they are. We talked about that actually. Um, Also, what's his name? Anthony Rapp. I didn't get to meet him, but I was like 20 feet away from him like a month ago at Awesome Con in DC. That was cool. Yeah, the whole tardigrade thing on the mycelial network. You know, right, because I don't know for sure. I really wish I knew who I could talk to. Actually, maybe through a sort of a moderate but growing amount of familiarity I have now with David Hewlett from Stargate. I wonder if he would be able to put me in touch with someone who wrote on Star Trek. I don't know if he has that amount of clout. But so with Star Trek Discovery and the tardigrades, their key thing was like, oh, tardigrades, they have like all this DNA from other organisms or whatever. So fun fact, that was a mistake. That was actually a mistake in the, look at all these diatoms. That was a mistake in the genome sequencing that was then published. They didn't issue a retraction, but they did issue a clarification. So when the tardigrade genome was first published, they saw a shocking percentage that came from other organisms, and they thought that it was through horizontal gene transfer or some other unknown mechanism. Turns out it was contamination, although they did go on to say, what is this? Is this a flatworm? They did go on to say that they still did confirm some exogenous DNA in the tardigrade genomes, but not as much as they first said. Okay. Discovery had its moments. I agree. You saw David Aishi's interview with Hewlett on YouTube. Seems like a friend of science. Wait, let me see. Aishi. I don't recognize the name. Knowing tardigrade researchers, they were all very disappointed when it turned out not to be true. Absolutely. Hello, what are you? Gastrotrich. Which unknown thing was that? I'll look it up. I'll look it up. I want to follow this little guy though. This is kind of a fun thing. He's not going to cooperate. Gastrotrich. Gastrotrich. What is that? Flatworm looking thing. Ah. Commonly referred to as hairy bellies or hairy backs. Cool. Oh yeah, you're right. 100%. Look at this. I mean, perfect identification. You got it. I feel like I should, there's got to be a way for me to program in. Oh, hold on. I'm back. Like community rewards, but more like rewards where I reward you when you positively identify something for me. Because I feel like I would be handing out tons of points today. Other friend was a rotifer. Oh, absolutely. I just don't know what kind. Wow, my fingers twitch. Oh, no, don't scare away the one that's not moving. Rude. Absolutely rude. Ah, and yet another thing. Okay. Okay. So, you can't really see it. Yes, that. So this little thing right here, I have seen these before. They are different than what we've seen before. These are predators. They hook themselves down and then they do this. They go in a searching pattern, actually. I know of a researcher that researches these guys. You can kind of see its foot right there. And they're reaching out to grab things out of the media. Rotifer. Swan tear. Is that what they're called? You are right. Lacrimaria olor. Lacrimaria. That's good. Yeah. See, by way of also introduction, so if you haven't noticed, by the way I talk, I am a scientist, but I'm definitely not a microbiologist. I just happen to own a microscope, and I love looking at things underneath it. I'm a genomicist and a molecular biologist. These cellular organisms are too big for me. No. Get rid of them. Just give me their nucleus and leave me alone. At least that's my attitude in lab. What's this little cross? Yeah. No, I figured that's what their name means. The lacra kind of gave me that clue. I don't know what this is. I mean, it's right next to a diatom. But are there star-shaped diatoms? Star-shaped diatom. Caltrop. See, though, are caltrops things? Do you work with yes, we use CRISPR quite often in the lab to the point where it is extremely routine enough that I was able to we did a mock experiment like a couple weeks ago here on stream, to like we faked, we did all the like planning stuff to like cause a certain mutation that that the science thinks is a way to prevent HIV infection in the CCR5 gene. But yeah, work with CRISPR all the time. Is Lacrima olor related to Dileptus? That's a good question. I don't know. Okay, if I wasn't following you before, you would now. Well, thank you. You can always unfollow then refollow. That might CCR5. So CCR5, I think, is a um, cell surface marker or cell surface protein. I forget exactly what its exact function is. Yeah, it's a chemokine receptor. Yeah, so it's a cell surface receptor that's uh, important in immunity, which is so that's unsurprising why HIV requires it to like attack cells. Uh, you think Christopher is the coolest thing to come out in forever? I, CRISPR. I do like Christopher, our friend Christopher. Um, oh, thank you, Dracula. Dracula? Not sure how to pronounce your name, but thank you for the follow. Uh, using voice to text while doing chores. Sometimes it autocorrects you. Yeah. Yeah, no, CRISPR is awesome. I mean, I love gene editing technologies in general. So I've talked about several of them, or about them several times. Ooh, look at that one. CRISPR for Dracula or Drac. That's easy to remember. Hey, if anyone's interested, actually, I should just might as well plug this since we have a couple, more than a couple new people. There is a Discord. We're generally quiet, but we share all sorts of sciencey things and pictures and ideas for stream. So it's welcome, or it's a welcoming place. You try to get your kids excited about that line of work, like science, like a career in like biology. If it's like a career in biology, I would that my entry to biology was multifold. Uh, it was science fiction, which I feel like should be kind of self-evident because I self-stylize this stream as partly science fiction based. Dinosaurs and insects. So, that trifecta was kind of the thing that did it for me. Ooh. Oh, big worm. Annelid. I'm shockingly not that excited about these big worms. They just don't excite me. All right, well, let's see what else is around here. Uh-oh. So this slide is coming to a close because it's slowly drying out. But what are these half circles? What are these? Oh, look at these little spirally guys too. We saw one of those spirals being eaten, sort of, by the Stentor. Oh yeah, rip Stentor, by the way. Like I can't get it back out of this slide. They're dead. Gene splicing in general, if you were their age, you would be using CRISPR every moment you could. Uh, yeah, I mean, Jurassic Park still fulfills that, I feel like, genetic editing and all that sort of stuff. It's very much in the popular media and has been for quite a while. Pretty diatom right there. Worms, exciting and new. No, worms are the opposite of exciting and new for me today. Sometimes I'm excited for worms. Today I'm not. I could become the high evolutionary. I should know what that's a reference from. Is that a Star Trek reference? You're making me look it up. Marvel. Oh, right. I'm a not a passing Marvel fan. I'm a light Marvel fan. Uh Doesn't this look a lot like Nostos? Ooh. Oh, this is new. Okay, I'm not really sure what's going on here, but I think I might zoom in really quick. No stock. I keep mixing it up. Yeah, there we go. Very, very pretty. No stock, no stock. I don't know why I always forget the word no stock. And there was something else right here. Not too sure what this is. Ooh. Little diatom going by. Oh, wait. This, I think it's either dead or dying, but I think this is a Daphnia. So Daphnia have this red eye, and maybe we're playing Spore for real. Yeah, a little bit. If it was Spore for real, it would have crashed 3 times by now. The last time I played Spore was like 2010, so I don't actually know if it's is it stable yet? Is it abandonware yet? Yeah, that's what are the diatoms eating the Daphnia corpse? No. Diatoms are photosynthetic, and this might not be a corpse. Is this a Daphnia? It doesn't look right, but it has the shape and like the red eye of a Daphnia. You can see right there. Maybe it's a young Daphnia. If it was Spore for real, we would have made a dick by now. Yeah, definitely. It's been stable on Steam for a long time. Oh, well, good for Maxis. Acroparis. They have a little beak and no antenna. Maybe. Oh, it's whatever it is, it's on the move. Actually, can we say none of the above? Now that it's moving, it looks not like any of those. Very strange. I don't know what this is. And this is what Discord is for. Take microscope shot, and now we have a record. While we're on the subject, anyone know of any other cell scale video games? I, in fact, do. A video game you might want to look at is Bibites. The Bibites. We actually had the creator of the Bibites on like a month ago or something like that. The Bibites. It is a cell-based evolution game. Let me grab the link quick. It's very fun. I've put it on stream before, just for fun. It's part game, part simulation. SimCell. Ooh, SimCell. I have not heard of that. That sounds good. And by good, it had the word sim and cell. 2 of my favorite words. All right. So this sample is coming to a close. It is drying up quickly. Very, very quickly. Flow technically counts. I don't think I've heard of flow either. Ooh. Yeah, you can see the water, the water kind of move it in here. All right, well, this has clearly been a super successful microscope stream. I'm so happy. It's kind of like it's almost as if this sample knew that I wasn't going to be able to stream next Sunday, so it's kind of given me all the stuff for 2 weeks for one. Yeah, a lot of interesting stuff to look at this time around. I am over time. I'm only over time in that I do have to work on my lectures for this week for class. So I do think I'm going to have to head out, unfortunately. And, sort of maybe not well timed, but, the sample's drying up. Why don't I just very quickly, I don't think that I will be able to find one, but why don't I just very quickly try to find a tardigrade? Because we have so many new people, and I've been able to be pretty successful with the tardigrades in the recent past, but I've let my samples, I've let them go for a little bit too long. So I don't think we'll actually find any, but I might as well look. Ew. Nice to meet me. Well, nice to meet you too. Yeah, the science Twitch community is pretty awesome. Thank you for coming. I appreciate it. And for sticking around at, after the raid. After being raided here. Oh, yeah, and Miss Rizzo, are you going to start streaming sometime soon? Because I'll probably have to raid someone in the meantime. But if you're looking for some, what am I trying to say? Alt. If you're looking for some co-work time, go hang out with Alt Miss Frizzle. Brand Rickman, new here, got recommended this channel from Scientist with a Laser a while back. Oh, that was very nice of him. Yeah, well, I'm glad, it's enjoyable enough to be recommended. So I'm glad you're here. I'm just looking for this tardigrade. I gotta find a tardigrade before I stop streaming. But these samples are awful. That looks like a dead Daphnia. They are infested with too much algae. Look at all this algae. That might be a dead tardigrade right there. I'm not going to bother zooming in. Yeah, well, welcome, Brand Rickman. I hope you had a time to at least, I hope you've been here for at least a little while, because unfortunately I'm just about to wind down for the day. If you can make suggestions as requests, can we get details on the microscope you're using in the About section? Yeah, absolutely. I can do that. But the only issue is that this microscope is a hand-me-down, a infinitely recursive hand-me-down. It's by a company that no longer exists and is extremely old. So I have found very little information about my own microscope, but I can definitely give the details I do know. Wait, actually, does my command CV say something about the microscope, or is that just about me? Blah, blah, blah. No, it doesn't. Never mind. Oh, you jumped on about 8 seconds before I announced I was getting off. Yeah, well, I'm still looking for a tardigrade, so I'm still on for another, little bit. Yeah, sorry about that. Poor timing. Wait, algae infestation. Yeah, I wasn't lying about the algae infestation. See, this is, whoa. Thank you for the follow, Ingenuber. I love that name. Oh, you love the command. Yeah. I have commands, but I often forget about them. You can also change my light color. That's a command that works. That's a regulars to the stream will know how fascinated I am by my own setup. So I have very recently added a bunch of chat reward or what are they called, channel point rewards, just because I love programming them in. I love learning by doing, so that's been fun. Yeah, absolutely. We can talk about epigenetics. Epigenetics is so much fun. That's actually what I'm teaching in class right now. All about epigenetics. My students are wonderful. They're all seniors in college. They actually care and they actually kind of get it. And look at all this pine pollen.
Did I jump scare you? Yeah, I bet I did. You coward. You make me sick. Your weakness is pathetic.
Yes. Thank you all, Miss Frizzle. Oh, that scared me. See, it's great because I like know that's there. It's like a threat to myself. Let's see. You knew it was coming. It still got you. I love channel point rewards. I love channel point rewards. Did I say that? I mean, I love making channel point rewards and then forgetting that they are rewards. I sound pretty legit. What's my PhD in? So my PhD, well, thank you. I'm glad I sound legit. My PhD is in biochemistry, but I more prefer just to say that I'm a molecular biologist or a genomicist because I don't like to do straight up biochemistry, old school biochemistry. What's with all this algae, everyone? New sample. But I like to do stuff with genomics. So what that is the study of DNA in the genome. So like how it moves around, how things interact with it, how it interacts with other things, how the underlying sequences are interpreted, that sort of stuff. Oh, I looked away and I missed the start. I missed the Enterprise. Yeah, so I hope that was clear. It's exactly what we need. I hope so. I hope I provide a service. A threat to myself, yes. It is a threat to myself. Yeah, so with the Star Trek one, I mean to add in some sound so I know to like look over. But to be fair, over on my screen that is my second screen, I have the stream up, which is on a little bit of a delay. So I actually did see it fly by. I saw it fly by and I saw myself not looking at it. All right, so I'm just zooming through some old samples that I have sitting here. This one is infested with just, wait, what are these? Just random single-celled organisms. There is no sound in space. That's true. But there's sound in Star Trek. Maybe. What are these? Thank you, Trainer Hinton, for the follow. Welcome. Yeah, there's a bunch of little... See, every single one of these is a little bit unique. Still no tardigrades. This is what, if I would have known we were going to have so many new people, I would have prepared a tardigrades like sample. I'll look through all of these. There's got to be some somewhere. I don't know what those little blobby things are though. So we've seen these on stream before. I do forget the names, but there's like microscopic, like almost like predators. They have like little spines that come out of them that they spear other single-celled organisms with. Those might be it, but I don't see the halo of the little tiny spines. So also maybe not. In space, no one can hear you say number one. I hope people have noticed that with the follows and the like subscriptions, their audio taken directly from Star Trek. There's also a raid sound that does not work. that is also from Star Trek. Ugh, get away. All right. Also no tardigrades. It feels like the early days of my stream, where it was just no tardigrades anywhere I looked. Ooh. Well, this one has a bunch of diatoms in it. And, hello, I believe that is a rotifer. You're not currently working towards a degree of any kind, but you mostly study physics and quantum physics and astrophysics, pretty much everything that deals with physics. Physics is something that I have to get people to come talk with me about, because oh, this is a tardigrade. Look at that. Now, she's a little dirty. She's in all this stuff. Why don't I grab her quick? We might as well get her on a slide. Although a slide is a death sentence, so how about instead I put her on a... No, I'll get her off. Our little tardigrade friend. Oh yeah, they're super active. They're very, very fun to watch. You know, why don't we dehydrate? Oh, that was another one. Okay, apparently I do have still quite a few tardigrades. Yeah, wow, a living one. Why don't we dehydrate one? Because then for next stream, I will have one on a slide ready to go. Because she'll be stuck there. They're always on the go but have no sense of direction. But they have a sense of purpose. Okay. Gotta get all my stuff ready. Let's see if I can do this. I got her, but I got a bunch of stuff too. Like junk. All right, there we go. Okay. So let's see. I'll see if she's on the slide. All right, abducted, yeah. Well, that's a dead tardigrade. Oh, I hope I didn't just kill her, because that's a dead one. But wouldn't it be extremely shocking to find out that they're mammals? Well, they're definitely not, so there's no fear of that. But that would be. They aren't insects either, but they're fairly closely related to insects. There she is. Wait, that's two. What? Did I get two? How many tardigrades did I get? I got so many tardigrades. Look, 1, 2, 3. Oh, they're playing. And they're so little. Oh, they're stuck. That's why. Yeah, they're part of like Ecdysozoa, so like they're technically related to crustaceans and arthropods and insects and all those sorts of things, but they are off on their own. And if it isn't sort of clear, because it probably isn't, all of my emotes are a tardigrade. Okay. I'm just looking around to see if how many tardigrades we actually got. And I'm also going to increase the light. So a huge downside to my microscope setup is that it is hot. Very, very, very hot. However, the nice thing about it is that when I want to dry out a slide, it becomes very easy. So as this heats up, this should sort of shrink back. So we just got to find those tardigrades. There they are. Who do we like the best? Which tardigrade do we want to follow? Let's do this one. She's a little bit more clean. Ugh. Let's also see if she's carrying any eggs. Oh, you can really see her little claws and all that. She's... Huh. So I think this one is maybe like not quite fully grown. Yeah, they did split from the arthropod lineage very early on. They are their own unique little things. She's very cute. Let's go find the other two. Yeah, these feel like they're a little bit smaller than the ones I'm used to. So they might be a different species than the like major proportion that I've seen. Or they are like not fully grown. I'm just going to zoom out a tiny bit. Tiny bit. Oh, wow. I said I was going to stop streaming like 45 minutes ago. That's okay. Where is the edge of the water? And how fast is it shrinking? All right. We might have to help this along a little bit. Yeah, I did. But then new people came. A raid happened, and I felt bad. And then we kept finding cool things and cool stuff to talk about. I don't feel bad. Where'd they go? Also, what's that? No time. Well, you're welcome. All right. I want to follow her. I do have some paper here to kind of blot the sides to soak up some water. I just want to be kind of careful that I don't soak up the tardigrades. Stream over if I do that. You also don't want to dry them out too fast. Yes. No, I do not. That is true. We gotta stress them out a little bit. Although they will dry out once the drying time comes. It will happen very quickly. Just check on that border. Yeah, so last time we did this, we managed to dry one out. It happened very quickly. And then like a week or two later, we dehydrated it, or hydrated it, because we thought it died. They will be warned by the change of osmolarity. Yes, it did come back to life. Yeah, we rehydrate. So it looked like just looked like it like smushed, but it did come back to life. It took a while. So apparently, the length of time that they require to sort of come back to life is related to how much damage is done to them as they are drying out. Look at them. So cute. Take a picture. I'm just going to sit here and drink my water. Something we were doing like a couple weeks ago too was we were trying to 3D print mazes for these guys. Yeah, good point, Micro. Yeah, coming back to life is the correct term because they die in definition. They go through something called cryptobiosis, where all of their life functions cease, at least as far as we can measure so far. It seems pretty clear that they die. If I ever download that clip and it isn't just on Twitch, you'd love to show your students it. Oh yeah, I can do that. Can I 3D print microscopic? Yeah, sort of. It's a little. It's not the greatest. I really need to change up my 3D printing setup to a different style than I have altogether. But I can use my local university's free print shop to do that. But I can print small enough that, yeah, actually we can get some pretty good like little channels. But like comparison-wise to the size of these guys, it's kind of as if you were, it would feel like you're in like a hallway that's like weirdly wide that's also like 20 feet tall. And that is the absolute smallest that I can make it. And even then, when we do print that small, it's very blobby. If you've ever seen 3D printing in person, you can see like the lines, but it looks pretty good. At that scale, it's like any error in printing is very obvious. If they somehow have a metabolism while at liquid nitrogen temperatures, then you'll be very impressed. That's true. I guess, so I was thinking more when they just dry out, but you're right. They do survive extreme freezing temperatures, so, they're dead. Your school blocks Twitch, but you watched a video about tardigrades, told a class about it, now they want to see it. Okay, I can work on that. I can try and find that. Remind me elsewhere, maybe, and I'll try and find it and take a look. Or, if we clip this, that will make it even easier once they finally do dry out. Okay, this feels very suspenseful, but why is this taking so long? Dry out. Wait, I thought they did survive like liquid nitrogen freezing. Oh, Ballant was saying that it's easy to make things for microscopic things to snag on items from filament printers. Yeah. Filament printers are great. They're just not the best. I see what you're saying, Micro. I'm misunderstanding the point you're trying to make, but you're making it. I'm not understanding it. All right. We're at another round of paper after I find them. Oh, actually, that side is much closer. Where are they? There they are. Where'd the other one go? The other sort of issue that we sort of didn't have was like them not being able to walk on glass. Bran Rickman, you've got to head out and continue searching out PhD programs. Yeah, it is a pain. Yeah, if you want any help on that, you should ask on Discord. We can at least point you to people. But yeah, you're welcome. Thank you for coming, and yeah, best of luck. All right, okay. I'm just watching my edges here. They are very cute. Very, very cute. Oh, great. You'll check out the Discord? Awesome. Yeah, Embus. I feel like they're cute because they're dumpy and they're goofy and they look like they like bumble around. That makes them endearing. All right, come on. See, now at this point, this little bit of water is keeping me from dinner, so. There was a third one. Where did it go? They look like mutant gummy bears. Oh, that's such a good, that would be such a good product. Like gummy grades. Tardigummies? I would purchase them. Do those exist? While we're waiting, I'm gonna search up gummy tardigrade. Gummy tardigrade. All right, they exist. Sort of. Oh, making gummy tardigrades. All right. Yeah. Fine. Not original. Everything's been done before. CBD tardigrade gummies. You know, I think a lot of people would purchase those. It's closing in. Am I at full strength? Hold on. Now I am. Okay. Ew, moss flavor? I don't think so. Not for me. No, thank you. Oh. So they're kind of moving, I think because it's suddenly beginning to dry way faster. Please, I hope so. I mean, I can actually see movement of it drying, so. I lost them. Where are they? There they are. Okay, so hear you out. Breeding project, make them hamster size. I think they'd actually be less cute because they would be very insect adjacent. And they won't survive. They also don't really have a face that you might enjoy. You know, I'm getting kind of worried that this big thing right here they'll like go to it. Uh-oh, they are. I need to move it. Okay, I'm going to ignore chat for a little bit because I got to move this big piece of something out of the way. Oh, no. I'm making it worse. No. I think I just ruined it. I ruined it. I tried to save. Oh, no, I didn't. No, I didn't. No, I didn't. No, I didn't. That's tardigrade. Watch it. It's happening right now. There it goes. And that was it. Oh, you can still see it moving. You could see it oddly dry out way too much. And that was it. Sorry that happened so quickly, everybody. Yeah, I did need something hydrophobic. I was afraid that as it sort of the water was going to come in, it was going to go towards that like algae first and then just suck them in with it. That's why I tried to move it. Yeah, so it was sort of retracting all of its little bits. Okay. So that maybe wasn't perfect. But that was good enough. So there she is. I do not know how I'm going to find her. There's probably another couple in there. In fact, I do see it right there. That's going to take a while. We'll just rehydrate the whole thing. So everyone remember where we parked our tardigrade. It's by this weird shape that I've been seeing this whole time. In fact, it's kind of an arrow. Okay. Need something hydrophobic, retracting its legs. Wild clap. Oh no, F. Can you mail them to people now? So, I'd love to. Yes. I'd love to be able to do that. I've thought about it. I've talked about it on stream. And the answer is probably. It's probably more of a hassle to like do all of this. Though. Oh, and Micro, you have even more information. The skin between its segments is going through a rapid change in water permeability. Very cool. Let me just turn this down. So that is that one. Yeah, we talked about bespoke tardigrades before on stream. Like I was thinking, like, what if this is like mounted now? If I got a teeny tiny pen, I could do, Dracula, like write it right above it, and then sort of cover it, tape it up, and send it off to you. I mean, give me $50 and I'll do it. But, that's a little tough. All right. Yeah, so for anyone who hasn't really been following along, this is called, or has been, this is called going into a ton state. Yeah. So it is very controlled. There's active research going on now trying to understand exactly the processes that are going on, how it survives, and what that might mean for sort of like, I don't know, how we understand how life works, how maybe we could utilize this in some of our own endeavors, etc., etc. It's also just really cool implicitly for the biology behind it, I think. Okay, but now it is an hour past when I usually start. I am now very far behind in my day's activities, but that's okay. This was a ton of fun. Thank you for everyone from the raid who's following, and if you just happen to stop in by accident anyways. Where should we go for a raid? Who should we raid? So both Science and the Paradigm Shift both dropped in, and they're both streaming. Alt Frizzle, you'll be for streaming later. Let's go. It looks like maybe the Paradigm Shift hasn't quite started yet. So let's go Science. Yes. Oh, you haven't seen Paradigm yet. Paradigm is just entertaining all around, and Science is science. What are we feeling? I'm feeling Paradigm Shift today. Oh, he is using the new printer. Okay, gotta make a decision. I'm gonna go with Science. I want to see if he's using, or they're using, their new printer. So we'll do that. Science. Yep, I see in the preview, new printer. We're gonna raid. Thank you everyone for coming. Next time will be on Wednesday. Probably talk about gene editing, science fiction. Maybe we'll do some programming to turn memes into genes again. We'll see what we do. But anyways, thank you for coming. And if you're new, thank you too for following. See you later. Have a good rest of your weekend. Bye.